Hi guys. I'm just on here getting ready to do this review. So I went through the test one more time and I apologize for being so late and getting it to you, but we're going to um, actually go through some of the pieces. So we're going to start with meiosis, mitosis. Um, if we are doing meiosis and mitosis, we start with one cell here. And so the first cell we're looking at is 46. That's how many chromosomes the human has. Sorry, it's kind of messy. When it's replicated, so when we're talking about mitosis, it's all somatic cells, cells that are not reproductive cells. And those are actually going to be duplicated into 92 chromosomes. So 92 chromosomes. Sorry, it's a mess. And so we have 46 chromosomes, 92 molecules of DNA. Once those are replicated, and this is during the S phase, so there's three different parts to the interphase, which is G1, S, G2. And so that part is interphase itself, and so replication happens in S phase. G1 is normal cell structure. Remember, G0 is where it goes out and it's kind of in its own little space, and then doing its things like not dividing anymore. So that would be like your um, brain cells or spine cells. So right now we're doing just normal, let's say, skin cells. So it starts with 46 chromosomes as humans have. They're replicated to 92. During mitosis, we have PMATs, we have prophase and metaphase and anaphase and telophase. Prophase is the prep for all of that. And so basically the molecules are condensing into these chromosomes and getting ready to prep. So they're basically just these X chromosomes within the lovely little cell. When we're talking about metaphase, right before it starts to divide, they're actually lining up in the center of the cell, getting ready to be split, where those lovely, this centromere is holding those chromosomes together. So we have 46 chromosomes, 92 molecules of DNA before we actually split up. And then during anaphase, remember they're so far apart that they're starting to pull together. And then when we get to telophase, telophase is when they're actually going to see a lovely little cell where all of the chromosomes are usually over on this side. And now we're going to see a little bit of cytokinesis where they're actually going in, and that's going to eventually come in. And this is going to be its own cell here. This is going to be its own cell here. And then for mitosis, we're done because it's actually separated. And at the very end, we have 46 here, and then we have 46 here on this side. So both of them have 46 chromosomes, and we're all good. Sorry, it's a little scratchy. Sorry, it's a little messy. It's not working like I thought it would. But then for meiosis, we actually start with where the goal is to go from 46 chromosomes. So we're starting with 46, and then we are going to end up having 4 cells, all of which end up with 23 chromosomes in it. Sorry, that's like not a 2. So 23 chromosomes because they are combining with the sperm or the egg or whatever the case may be. The difference between meiosis and mitosis is this has 2 divisions and it also has 2 unidentical cells. So they're basically genetically different. Genetic difference. They're different. Sorry, this is not what I was hoping it would look like. So when we're looking at that, you're ending up with 2, you start with 2 cells, 4 cells that are basically genetically different, and with a lot of genetic variation. We talked about this last night. So this is the ultimate goal of meiosis, where the actual goal of mitosis is the 46 and the 46. We end up with both of those cells. These are identical to each other. All of these are identical. These are genetically varied. So this is meiosis. Again, those cell pieces are there. Make sure you know interphase. Make sure you know the pieces of interphase. The differences between what we end up with here versus this. The paper that I gave you guys last night where it's asking how many divisions each one, the comparison to contrast between meiosis and mitosis is a great way to actually get the idea of how this is going to work. So I'm just going to erase that really quick. Okay, so another thing that you're looking at for the test is making sure that you recognize the non-template and the template. Remember that a template is the opposite of the 5 to 3. We can only read DNA at 5 to 3. And so make sure that when you're reading the DNA that it's 5 to 3. We're not reading off of the template strand. The template strand is the opposite part. So if we're reading off of the non-template and we have, say, CAC, sorry, and it's running from 5 to 3, we can use that. If we had something like this, where CAC, TAT, oh, so bad. So you would have CAC, which would be the same. So because this would be the non-template, we talked about that before. The non-template, you can just take these guys and change out the T's to get to the U. So that your RNA strand, so this is all DNA because it has thymine in it. Your RNA strand, if it has the non-template, is CAC, right? And then we can just exchange these because we're working with the non-template. We can exchange it to the U, to the A, and to the U. So we basically are just switching out the T's. If we're working with a template strand, then you have to go backwards and do a template strand is usually 3 to 5. So you have to go back and redo the template to make it a non-template, and then we can make that adjustment through the DNA. Also making sure that if there is an anticodon, that it is the opposite. So the anticodon is, they're also read 3 to 5. So when they're clicking in, remember this is what's on tRNA. So the anticodon is 3 to 5. So the corresponding one that you need is 5 to 3. So if I had per se like a C, that's my anticodon. That's the one that's on the tRNA, that's dropping off the amino acid. This is our mRNA. This is the one that the ribosome is using or reading. And my codon on the mRNA would be UG. So then you would look that up, and this is what would give us what the amino acid is. So this is basically our transfer, our transfer RNA, and then our amino acids up here. So whatever that amino acid is, this is the ribosomes. This is what we're reading. This is the mRNA. And then this one is tRNA. So it's actually the opposite. It's running 3 to 5. So tRNA is my anticodon, mRNA is the codon, and of course it runs opposite. So make sure you're aware of that. Make sure that you can read a codon chart. Make sure you understand how to get the amino acid based off of that. Also make sure that you understand the differences between a missense and a nonsense, and the other type of, sorry. So remember a missense, if we're doing missense, the missense is when it's looking at a different amino acid. Amino acid is different. So this is the amino acid that comes up differently. And then the nonsense is our lovely stop codon. Okay, so if you have the missense is the amino acid, is a different amino acid. The nonsense is the stop codon, and then you have the silent, which is basically one code off. So it's this one generally will have the same amino acid, but the code itself is different that it's calling for. So make sure that you could recognize those. As far as the being able to do mitosis, meiosis, if I have a cell that looks like this, and they're all lined up like this, make sure that you could recognize that this is, say, metaphase, right? Because they're all lined up on the plate. The way that I remember this is prophase is the prep, metaphase I met at the middle, anaphase I moving away, and then telophase I'm so far away that I have to telephone a friend, and cytokinesis it comes in and cuts the line. So hopefully that will help kind of remind you of what you're doing during that time. Make sure that you are looking at how the structure of a homologous pair looks. So for example, if I have a homologous pair here and a homologous pair here, and I'm talking about meiosis, we're probably going to see what we call crossing over, and we talked about that this time. So let's pretend that this is all red. And then this guy over here is a blue. Kind of looks like that. Crossing over. Make sure you can, so this is mom's chromosome that she gave to you. This is dad's chromosome. Crossing over is when they actually exchange DNA. So that's one way that variation happens. Then we're talking about independent assortment, which means if we have a cell that's lining up, they don't line up dad to mom on this side. So not all these guys are dads and these are all moms. They kind of go back and forth. So we could have some mom's chromosomes on this side, some of dad's on that side. That's independent assortment. And then the last one would be your variation and genetics mutations, that kind of stuff. So this is what we covered last night, those three things. Sorry. Not my favorite way to do a chalkboard. I'd rather be writing it. So make sure that you understand when sister chromatids are separated out. So remember that when we're looking at a chromosome, this whole thing is a chromosome. But these guys, so this is, let's say this is our 1/2, and then 1/2. There's a chromosome here, a chromosome here. In the middle is our centromere. That's what's holding it together. That's what tells it that it's one piece. So this whole thing is one chromosome, but 2 molecules of DNA. And so the centromere is what's going to be holding it together. And then when they are pulled apart, when both of these are going in this direction, when we're in either mitosis or meiosis, remember that in meiosis, they're being separated during anaphase, right? And which pieces are doing what. And that all goes back to being able to recognize the steps of the steps of PMAT and how that works. Also, make sure that you remember that spindle fibers are the one that's going to come in here and then pull those apart. And also that belt, the seat belt that basically clicks, the spindle fiber clicks into, is called the connecticore. So that connecticore is basically this is the seat belt into the belt, kind of the cable into the belt. And the connecticore is what's holding it, or what's going to be able to pull it apart, so that this, again, this chromosome goes that way, this chromosome goes that way. Make sure that you are familiar with the replication of DNA. So Replication of DNA means that we have ligase, right? We talked about this last night. And so you have ligase, you have RNA polymerase, you have DNA polymerase, and I put poly, so sorry, it's not that great. RNA and DNA polymerase, ligase, and then you have your primase. So what each one of these are doing during the time that DNA is being replicated. So ligase, oh, and then there's your helicase. Okay, so the helicase will come in. Make sure you know the difference between them. So helicase comes in and it opens up the DNA, and then the primase will tell you where it's going to start replicating. So we have DNA that's split. Our helicase comes in. Our primase comes to here. It says you're going to start replicating here. And then DNA polymerase will come in and basically build the strand that's being created. The RNA polymerase will go in and double check, and then ligase will basically glue it all together. So now you have a nice little strand of RNA to go out to the ribosome to be able to make amino acids. Okay, sorry my pictures aren't super great. Let's see, a couple of other things. Make sure you know the cell cycle. And then make sure you understand how cancer works. Go back in and look at those cancer pieces and what's entailed in that. So like P58, that particular negative feedback piece that allows the tumors to not grow. Make sure that you are looking at basically variation in how evolution takes place, mutations, and how some of them can be pretty harmful, helpful. Apoptosis, we talked about that, where it can be very, very harmful, but it also is very helpful if I want to have like 5 fingers. So make sure that you understand the difference between mitosis and binary fission. So like, for instance, bacteria uses binary fission, and skin cells use mitosis. So there's a big difference. And then cytokinesis, make sure you're understanding when that takes place. So cytokinesis basically takes place after the division of the cell. It's the final step to basically pinches things off. Be very familiar with the differences between what's going on in each stage. So prophase versus metaphase, what the chromosomes are doing, what they look like. Be able to identify that in a picture. If I gave you a picture of, say, chromosomes at the very end and the cell starting to look like it's splitting, that would be telophase. It's not quite cytokinesis until that cell is split. And remember that cancer cannot be a result of one single mutation. Remember that there's several mutations that are involved in that to have cancer actually be evolved. And then make sure you again go back to look at those signal transduction pathways that help or cause a cell to actually divide. P53, that negative regulator of the cell cycle, make sure that you recognize that as responsible for arresting the cell cycle and how that works. And then the difference between a somatic cell versus a reproductive cell. So somatic cell is basically all cells but reproductive cells, and their number of chromosomes is 46. If it specifically says a reproductive cell like sperm or egg, then now that chromosome number has been halved. Haploid is half of the chromosomes. Diploid is all of the chromosomes involved. So a diploid number for humans is 46. The haploid number for humans is 23. And then remember that not all species use X and Y in order to determine sex and how that looks. Let's see. Again, make sure you can explain the difference between nonsense, missense, and um, single mutation. So nonsense would be your stop code in the wrong place. Missense would be additional amino acids that are built, not the right ones. And then our silence is that there's a mutation, but it doesn't change the amino acid in the backside of it. I've gone through this a couple of different times, and this is a little bit of helpful. I know that it's not as nice to have the board behind me, but I'm trying to give you all of the vocabulary um, that I can give you technically. Again, if you have questions or you have that you're not able to understand, you can always text me. We can Zoom. If you need it desperately. I have a couple of available times, maybe tomorrow even, but this is most of it is DNA replication and how that works. All of the pieces that go with that. So DNA, RNA, polymerase, ligase, um, helicase, how that all works. And then you want to make sure that you can read codons. So where how many codons are in each one? There are 3 bases, so like ACC. And then we're reading DNA from 5 to 3. And if it's 3 to 5, we have to flip it and make sure we're looking at it. Make sure you know the difference between tRNA and mRNA. tRNA will be used as the anticodon, which means it's the click in. They are read from 3 to 5, where mRNA is read 5 to 3, just like DNA. Every base for the codon, there are 3. Remember that the bases are different for DNA versus RNA. DNA has a TCG, where RNA has, did I say that right? DNA has a TCG, and then RNA is a UCG. Apple under the tree, car in the garage. That one is RNA. Apple in the tree. Apple in the tree, car in the garage is DNA. Apple under the tree and car in the garage is RNA. Hopefully, I know this isn't super helpful, but I hope that this is better than just kind of blindly studying. I've looked through all of the questions. Make sure you know specifically how the cell division works. That meiosis is 2 divisions. You end up with 4 haploid cells in the end that are genetically different. The one crossing over takes place in prophase, and there are 2 cells after the first division in meiosis, and then you go to 4 haploid, which is 23 for humans. For mitosis, it's just normal cell division, so it starts in prophase, again, prep. Metaphase, they meet at the middle. Or anaphase, they're moving away from each other. Telophase, they're so far apart they have to telephone a friend. And then that's it. You end up with 46, 2 cells of 46 at the end of that process. I highly recommend that you go back through and look at the PowerPoints. I posted all of them for the entire unit. And I definitely would say that those lovely little extra quizzes are super helpful in order to get ready for this test. So make sure you understand where all the checkpoints are for the cell cycle. So checkpoint 1 is at the end of G1. Then you have checkpoint 2, which is at the end of G2. It's basically checking to make sure all the DNA has been replicated. And then the last checkpoint is in checkpoint at M. So it's right at metaphase, just making sure that those cables or the spindle fibers are connected into the connected core so that they can split those chromosomes evenly and that they don't go one way or the other. Again, I hope that this helps. I know it's a very short, I try to keep it short and sweet, but there's really, most of your test is just knowing how to read codon charts and just the bits and pieces, some of the vocabulary of meiosis and mitosis. A large majority of it is those vocabulary words, and then DNA replication. So again, there's also a couple questions on cancer, and are cancer mutations all the same for the same type of cancer? If I have pancreatic cancer, is it the same type of mutation for all of pancreatic cancer patients? No. Um, so just keep that in mind. And again, if you have questions, you can email me through Canvas. I have my phone with me all the time. So if I see a question come through and I'm doing something, I'll try to answer it as quickly as I can. And then again, if you have other questions, yeah, just let me know. Make sure that you study the vocabulary a lot. And by vocabulary, I mean like what is a chromosome, what is a chromatid, what is a connector core, what is a spindle fiber, how do they work inside of these processes. The end process of meiosis is those cells. The mitosis is genetically the same. All cells are done by mitosis except for those reproductive cells, which are done by meiosis. So again, I know you guys are going to do great. I hope you have a wonderful weekend, and we will meet back up on Monday. So have a good one. See you guys next week.